shift the gears of the conversation a bit, and we would like to ask you that like what did you do in your PhD in terms of the scientific questions that you asked, and like how did you try to answer them?
Okay.
Just shift.
So during PhD, I have worked with the dengue virus. Dengue virus is one of the common pandemic that is happening in the tropical countries, and it is a mosquito-borne disease. So, as the mosquito is present, the chances of getting dengue is also high. So, and the severe cases of dengue every year it causes many deaths. So, it is very very important to study the severe cases of dengue. So in my study, what we have done is we have checked how the vascular permeability happens during dengue infections. So during severe dengue cases, we have seen, I don't know whether you have seen it, many people have rashes on their hands and all if the dengue fever is so high, like the severe cases of dengue. With this rashes is actually the vascular permeability, and this can lead to dengue hemorrhagic fever or dengue shock syndrome that can lead to their death also. So we ask the question why it happens, why the vascular permeability happens. So what is vascular permeability? Vascular permeability means it is the leakiness of vessels. So in a normal condition, the blood vessels are tightly packed, watertight, so that none of the components will ooze out from the blood vessels. So here, when there is a dengue virus infection due to many different signalings, so the patient may get the vascular permeability. So to identify the factors of related to this, what we have done is we have infected the endothelial cells, because endothelial cells are the part of the this blood is responsible for the vascular permeability. So we infected the endothelial cells with dengue virus, and we checked them for different expressions of the proteins, whether some of the proteins are upregulated, means increase in expression, or some of the proteins are downregulating during dengue virus infection using the transcriptomics method. Transcriptomics means it is the measure of mRNA copies of a gene. So that if the higher the mRNA copies means that gene expression is very high, or that gene is upregulated. And in our study, we have seen that there is a particular signaling pathway molecules are getting upregulated, and those molecules are related to the vascular permeability. So this one, we have analyzed it and we validated. Just seeing the transcriptomics data won't give us to validate it. We have done the qPCR and the immunofluorescence assay. qPCR will give us the idea about the gene expression. Immunofluorescence, we can see the protein expression. So we have done this to validate our study. Yeah.
It sounds like both your PhD and your postdoc work revolves around understanding how viruses hack our biology. So, what drew you to this area of research? What was your motivation?
So, the passion for biology started for me when I was in ninth standard, where we study the Mendelian genetics, and from that, when we go to the higher studies like the plus two that 12th standard, it deepens my passion for biology, and then I took the future path as biotechnology. So here, when it comes to virology means it is not the study of viruses alone. So viruses are very minute, very small. We can't say living organism, but when it comes in contact with the cells, it is a living organism. So it gives us a better understanding of the life, how the living organisms work, and they are very cleverly hijack the host system. So we are able to understand the molecular mechanisms that is happening in the proteins involved in the host system. So in that case, so virology will help us to understand the how the proteins, how the genes are expressed, how they are turned on, turned off, how the proteins are made. Then also we are able to understand how our immune system responds. So how can we edit the genes? The CRISPR is actually discovered from the virus defense system in bacteria. So in my PhD work, my work focused on basic research, and in the postdoctoral work, I'm focusing on the advancement, like the technological side. So virology has different realms. So that's why I am interested in studying the viruses, because we could understand many things that is happening in the host cells.
So, like, every virus might have a different strategy to hack the hosts, and I think it would be very interesting to see how they work. And I mean, like, at any point, have you felt like, okay, this is turning out to be more complex than originally thought, or like, do you like enjoy the process of it all?
Yeah, actually, we even though we say viruses are very small, it's like very tiny organisms. But the thing is, like it, the way it infects is really complex. So each viruses, like each family of viruses, infect the cells in a different way. So if we study one particular virus, doesn't mean that we can reciprocate in another one. And we have seen some other virus, some other scenario, like one protein of the host cell might be antiviral to one particular uh, virus, but it is pro-viral to another virus family. So it is really complex. And understanding this, actually I enjoy this. That's why I am in this research.